Dr. Lynn Rothschild she's a renowned astrobiologist and synthetic biology. She currently works at NASA Ames Research Center. She received her PhD in molecular and cell biology and her postdoctoral, and both from Brown University. She currently serves as adjunct professor at Brown University and has previously served as adjunct professor at Stanford, where she has taught astrobiology and space exploration for a decade. And in both those roles, she has served on multiple faculty advisor to student projects, specifically the Stanford Brown iGEM team. As you will see today also, she is a passionate astrobiologist, focusing on the origin and evolution of life on Earth and elsewhere, while at the same time pioneering the use of synthetic biology to enable space exploration. Her research focuses on how life, particular microbes, have evolved in the context of physical environment, both here and potentially elsewhere. She was awarded the Isaac Asimov Award from the American Humanist Association, the Horace Mann Award from Brown University, and has been a NASA Innovative Advanced Concept Fellow 5 times. So I'm very, very happy that she could join us today. And her talk is on Star Trek Meets ET, synthetic biology as an enabling technology for space exploration. I've listened to her talk 2 times before. It's amazing if you get a chance to listen to her.
I'm hoping I don't have to do an interpretive dance.
So, if you can join on Zoom probably, and you can connect my I've never seen it. Does that help?
Yeah, I mean, I'm picking up do we have a way to explain it?
Can you project my computer?
Why not project my computer?
Yeah.
Just tell me what the room is. That would be that, yeah.
You can set it up right in the phone.
So my file that I've never seen, I was like,
I'll send you the. Ah, okay. Okay, let's start again. It's usually at this point in the talk that I explain that I'm not for the technical end of NASA, I'm a biologist. Somehow people expect if you're a NASA employee that you can, just sit down in the morning and board a spacecraft to go to Europa, but I can't, obviously. But anyway, through the powers of technology, we've done this, and only once I've had a total failure to a bunch of docents from the California Academy of Science, and I did really, literally do my talk sort of as an interpretive dance. You do not want to see this. So anyway, thank you very much for coming. I give lots and lots of talks all over the world, and I have never seen a departmental seminar at 9 a.m. And I'm shocked that there's anyone in the room. So, thank you so much for coming. I really appreciate that. And so let me just explain a little bit about my background. I'm a protostologist by training and by love. I love algae, I love protozoa. I even love red tide. I worked for my master's in organisms related to those. But so I ended up at NASA for a postdoc and got very heavily involved in astrobiology and founding the field, the search for life in the universe. And my lab still does quite a bit of that. But about 15 years ago, we had a center director who was an astronomer, but became very excited about the potential for synthetic biology to help get humans off this planet. And he put me in charge of it, and he's a retired general, and I do work for NASA, so when you're told to do something, you sort of have to do it. And that's absolutely changed my life. I wouldn't be here otherwise. And so what you're going to hear today is really that side of my life, and most of my lab at this point is focused on that, although, again, we sort of mix it with the astrobiology, search for life in the universe, and all that sort of fun, alternative talks. So anyway, I fancifully called it Green Continents for a Red Planet because I thought that was kind of fun. So I don't know if we can turn down the lights so that I can take you off planet and you can sort of have your out-of-body experience. This talk is going to be incredibly low on data and incredibly high on ideas and excitement. And so I want you to be in the mood here. So here we are on planet Earth.
And here we are at NASA Ames where I work. We're actually one of the first centers. We predate the founding of NASA. We're having our 83rd birthday, I think, soon. NASA was founded in 59. So here is the Moff Field Park. My office is way down there. The big hangar there held the Akron and the Macon, the blimps, during the Second World War. It's being refurbished because of asbestos and so on, and that's why it looks kind of skeletal. And so if you communicate to your friends or neighbors on planet Earth, it's very simple. You don't even notice the time gap between when the words come out of my mouth and when they enter your ears, and certainly not the visual. But as we start to get off planet, here's a Landsat image of the San Francisco Bay, and I've put NASA there so that you could find it, and you can certainly find Stanford there, and San Jose. It becomes more challenging to communicate. And as you get even further, and here's a picture of ISS, the challenges become increasingly large. The communication to ISS is not too delayed, but then you have the issues of the material transfer, getting up resupply of people, clothes, food, et cetera, et cetera. As you get to the moon, it becomes even more of a big deal. Looking around the room, I may be the only one in the room who remembers Neil Armstrong stepping on the moon. I was pretty young, but I did stay up all night watching. It was really exciting, and yes, I have met Neil Armstrong. But everyone in this room should also have that experience again within the next few years, and it will be amazing. Much as I believe that robotic missions get you much more scientific data, there's something unbelievably exciting about seeing a human step on another world. And then when you get to Mars, you've got even bigger delay. It only takes a couple of days to get to the moon. It takes, we figure at this point, a nominal mission would take about 6 months to get to Mars. You then have to be there for about 1.5 years till the orbital mechanics allow you to have a short transit back. So you're talking about a 2.5-year nominal mission. And Earth becomes even more remote, and you have to become even more self-reliant. So I'm going to ask you, can you find Earth in this picture? It's, boy, eagle eyes down here in Florida. Excellent. And I mean, just because I can, here's a Cassini picture from the Saturnian system. Can you find the Earth? Very good. That's the little dot that's us. And so, you know, as they say, everyone you've ever known, every person who's ever lived, every, so on and so forth, all tied up in that little tiny dot. And so this makes the point that we need to be self-reliant as we go further and further away from Mother Earth. However, humans have needs, and it doesn't matter whether you're on Earth or off Earth. We need transportation. We need life support, we need food, we need oxygen, medicine, waste recycling, clothing, and so on. Now here, oxygen is a given, but that's not true when you're off planet, and so suddenly that becomes really important. Habitats, you need power, heat, light, you need radiation protection, which again becomes even more important off planet as you go away from the both the atmosphere protecting you from ultraviolet radiation as well as the electromagnetic protection that we get. So we really need to worry about radiation protection off planet. We need communication, and because this guy's waving a lot of cash, remind you that, as they say, there's no bucks, no Buck Rogers. So those are all the needs for humans, and as I said, we need those whether we're on planet or off planet, but we at NASA and other space agencies have certain challenges that are somewhat different from an Earth-based system. And one of the biggest challenges is up mass, or up volume. And this is basically the idea that it costs a lot to take something off of Earth's gravity well. Much of the fuel that's used in a spacecraft is burned in the first few minutes just to get out of Earth's gravity. And so anytime that you can reduce this up mass, you increase your mission capabilities because then you can take other things with you. And so whereas if you order a 100-kilogram piece of equipment for your lab bench, you wouldn't think anything of it. But I might want that at 3 grams. And so there's this enormous push for miniaturization and automation and being very clever. And if you can do things off-planet, even better. And so this is, again, tied in with the cost. You also need things to be able to be stored, they need to be reliable, and they need to be flexible. You cannot just go to the moon or Mars and say, oops, I forgot a hammer. You can't just call the hardware store or Amazon and get a new one. You better be able to deal with what you have, and ditto for food and medication and habitats and all these sorts of things. And obviously you need to worry a lot about the source of power. And so I've started adding in this last little bit here that really in many ways this is the ultimate self-sustainability circular economy, because you can't cheat. I don't know how many of you are aware of the Biosphere 2 in Arizona, and it was a great effort, and it's still certainly ongoing, but ultimately they cheated because they, the human life came first, and they did, let in the oxygen and bring in a few more materials. You can't just decide one day that you're going to cheat off planet. Oh, I'll just go down to the hardware store, because there isn't. So you really need to know where everything is going, including to some extent every car. And so what I've been selling for the last 15 years is the idea that we have a technology that can solve a lot of those problems. So what do I have in this black box? Well, it's a technology that's programmable, and that's, in this day and age, a given. It's modular, yeah, that sounds great. Self-replicating, and that's when the engineers sort of go, whoa, self-replicating, and then you have visions of self-replicating cell phones and all that. And better yet, it's self-repairing. And now you're saying, gosh, this must be a really, really good technology. And by the way, it has all these other capabilities. And so you're saying, wow, I need this technology now. And of course, what I have just sold you in this black box is life itself. And as I said, I am a biologist by love and by training, and so I'm a scientist looking at the beauty and the diversity of the natural world, and now I'm flipping around and trying to think of it as an engineer, and I know many of you are from more of an engineering background, and you're suddenly saying, whoa, this has all these cool capabilities, what can I do with it? And so I must say, this last 15 years has sort of unleashed my inner engineer. And so here's a sort of an unnatural vision for me, but a great vision, and that is looking at life as a micro machine that does carbon chemistry and nanotechnology. And once you acknowledge that, then the rest is like amazing. So what's the big idea? And this is why I often call this talk actually from Star Trek to ET, because it really is sort of this fanciful stuff. So what's the big idea? So let's start with a problem. Now we need materials off planet, but if we bring them, it adds to the up mass, and as I keep saying, it decreases the mission flexibility, because if you're going to bring a habitat, that means you can't bring something else. And you also have to worry that a lot of materials aren't stable, and so on and so forth. So this is a big problem, materials off planet. So what's your solution? Well, why not use life for material production? So, for example, life is a material. We use wood, leather, cellulose, and so on. That's why I go around Scotland taking sheep pictures whenever I'm there, just to remind you. And of course, you can add bioprinting in and make all sorts of amazing things. And I wish I had time to talk about that project, but I don't. Life also makes materials. Now, I sort of split the difference here. For example, it produces chemicals, and I'll tell you a little bit about our astropharmacy project in a moment. And it also can bind other materials and make biocomposites. And so this has been one of our big projects that I'll tell you about in a second. But the problem is that most of these things that we use for making other materials in the earth, for example, when people take trees and they make nanocellulose, or they strip down feathers for keratin or whatever, we're not going to take chickens, and we're not going to take sheep, and we may take earthworms, but we're not going to plant cotton fields. We're not going to do all these things off planet, at least for the foreseeable future. But what we are going to take is something like this. And this particular bacterium is Bacillus subtilis. And the reason I'd like to show this picture is you can see all those white dots. Those are spores. Bacillus subtilis is a common soil bacterium, but it's really good at forming resistant spores. And when I say really good, the spores from Bacillus subtilis were flowing on the long duration exposure facility for nearly 6 years. They didn't really plan on being nearly 60 years, but it was, the recovery was delayed. So we have nearly 6 years of data, and many of these spores survived. So when I flew a payload on a German satellite a few years ago, I flew Bacillus subtilis spores. It's not because they're particularly beautiful or I have some passionate attachment, but as I always tell people, it allows you to sleep at night because they work. And so I would strongly urge people to use Bacillus subtilis spores as the form factor to take all these capabilities. So you start to think of this natural world as this giant hardware store of biological capabilities that we then package in a Bacillus subtilis, put in the spore form, and ship it off planet. And now you can make things like spider silk or deposit silica or make cellulose and all those sorts of things. And so that's the vision. Now, on planet Earth, the way things work, and this is really, really basic ecology, is we have carbon dioxide in the atmosphere. Even though we're very concerned about it going up, the actual percentage is infinitesimal compared to Mars, which is 95% CO2, for example. Well, you take the CO2, we take light, we take water, we take minerals, and autotrophic organisms, for example, grass or algae or whatever, take this and provide that interface between these raw materials on the planet and organic carbon, which is then available in things like sugars and other organics for the secondary producers like a cow to eat. And then sort of an abbreviated ecological table, we could then take these products and use them for food, fuel, clothes, drugs, habitats, and so on. And I'm suggesting that we do the same sort of system off-planet, where we take organisms to provide that interface between the raw materials that we have. We'll still have the sunlight, we'll have the water, there is some water on the moon and on Mars. It may not be liquid, but we can always melt it and put up pressure. Carbon dioxide, much more on Mars than the moon, but we will find the CO2, there certainly is some on the moon, and the minerals, and maybe use bioreactors with photosynthetic organisms that then, again, produce the organics, because even though there's some spurious reports of organic material on Mars, it's not the sort of level that we would really need to do anything. We're going to have to produce our own. And then maybe we take those and then we feed them to a Bacillus subtilis that's been programmed to make, spider silk, cellulose, drugs, whatever, and we're good to go and potentially add the bioprinting. So that's my vision of how we're going to do these biological economies off planet. And so what I'd like to do is give you example projects that we've done over the years. Habitats are certainly a big issue. Here's a picture of the Apollo 11 lunar module. Now, if you're the first person to step foot on the moon, you could probably live in darn near anything for 36 hours. And that LEM was extremely small, as you can see, and cramped for 2 people. Now, I did a little calculation. The internal volume of the lunar module was about the same as the C block cell in Alcatraz if it was only 5 feet high, so not even my height. And I'm sure no one has been incarcerated in Alcatraz in this room. Maybe some of you have visited it. It's a notorious prison off the coast of San Francisco. And you can imagine these are not luxury quarters. So again, the point is that this is a very tiny, cramped place. But if you're only on the moon for 2 nights and you're the first people or even the 10th or the 20th, yeah, okay, I could deal with that for a very short period of time. But you cannot expect people to live in quarters like this for long periods of time. And so not only we have plans for bringing habitats, for example, to Mars that are 29,000 kilograms. That's quite a bit of upmass. I mean, everything's there. It's a nice sturdy structure and so on, but it's a lot of upmass. And so we ask ourselves, is there a better way? Because you can see there's a lot of material on the surface of the moon and Mars. And so some of the ideas that have been batted around have been robotic construction of regolith or ice houses. Regolith is just that surface material. We don't call it soil because it doesn't have organic carbon in it. Or what we've suggested over the years is to turn to a mushroom. Well, actually, more to the point, most fungi have pruning bodies called mushrooms that you're well aware of, but they're actually filamentous. The main body of the fungus is this mycelium, these hairs, this hair-like network that's underneath the surface. And others have discovered that if you take the mycelium, it's really good at binding other materials. So, for example, you can then take the mycelium, put it in a mold, and put in wood chips or garden clippings. Unfortunately, lumber yards and garden and lawn clippings are also not abundant on the moon or Mars. People say, oh, these are cheap materials. It's like not actually my biz. But anyway, you can use them certainly on Earth by putting them in a mold with these things, and then they bind them, and then you bake, do post-processing of just literally baking them. And you can get materials that really are very, well, basically indistinguishable from particle board. I did not bring one of my bricks with me because it's sort of heavy to carry around, but it really, it looks like and can feel like particle board if you're binding wood chips. And I tell people, here, look at this, and they say, oh, it's particle board or harder. And I say, smell it, and you smell it, and it has this sort of vaguely mushroomy smell. It's really quite fascinating. And so there are multiple uses. It's sustainable. Actually, they're very good acoustic properties. There are a lot of really great properties, insulating and so on. And think about it, in principle, you can just compost an end of life. And so this is a chart from a recent paper, and I don't know if you want to bother reading it, but it does show that the strength is actually quite good for the sort of mycotection that we're doing way over on the right. And the heat losses U value is extremely good. And one of the really cool things is that you can make this at room temperature, basically at 20°C. And so there should be a lot of really great uses. So we have had this project from NASA. We've gone phase 1, phase 2, and we're towards the end of the phase 2. We would love to get a phase 3 and take this actually to the moon. This is the vision that we have that was drawn up by the main architect on the team, Chris Maurer, the Red House Studios in Cleveland. And let's see if it'll play. He put together a little video that sort of explains our vision here.
In the near future.
I don't know if you can hear this or not,
Yeah,
but it doesn't say anything that you can't read.
A hermetically sealed bags delivered that contains microbes and spores.
And this has to be hermetically sealed because of the atmospheric pressure being low there because of planetary protection
Nitrogen
considerations.
from Mars. The form fills with air and water, and the embedded algae grow. The reaction creates oxygen and biomass to fill the form. Fungi are released, which bind the dried algae biomass to create strong composite. The fungi can be made to secrete enzymes and polymers that have myriad benefits.
Obviously, that last image is a little fanciful. We do not expect there'll be women walking around with skirts carrying handbags. But it does point out that there are two companies in the East Bay and San Francisco who are working on building, they can't say this, but I can, basically imitation leather. And I've seen this. So rather than using the fungal mycelium to bind other materials, they just use it as is and then tan it. And both of them, because they're private industry and not government, they're out to make money. And so both of them are going for the high-end handbag industry. So if you see these high-end handbags that are advertised as sort of vegetable leather, obviously fungi are not vegetables, it may well be one of these mycelial things. And I've seen them. It's really remarkably good in terms of being an imitation leather. And you, as you can imagine, you can't build a real leather off planet unless you have a cow with you or a sheep or something, but you could build a mycelium one. So you could, in principle, make that handbag she's carrying. It's just no female astronaut's gonna. As I mentioned, we have to do this all in an enclosed closed structure because the pressure is too low on the moon and Mars, and when you get to Mars, you have planetary protection requirements, which means you can't release Earth-based organisms, just in case there's any chance that there's an indigenous biosphere. And sort of my biology hat, if there was a second form of life on Mars and we destroyed the potential for finding it because we were sloppy, that would be one of the greatest scientific tragedies of all time. So we have, as a member of the team, someone who specializes in building inflatables for off-planet. And so he built us a little inflatable so that we could prototype it. And obviously this is a time-lapse image. This is not how fast it would go. But this has been sort of my image of the outside for this sort of habitat. And let's see, we've tried growing these fungal mycelia on basically everything in our pantry, and the students have grown it on basically everything, including, really importantly, on lunar and Mars Regulus simulant that had some nutrients added. So you just take a little bit of a liquid medium and just put it on this sand material that looks like sand, and the fungi still grow on that. And so that's really, really encouraging, because there aren't garden scraps on the surface of the moon or Mars, but there certainly is plenty of regolith. So when I tell people these are great, they literally grow on garbage. They do literally grow on garbage. And so my students, this is work done by the Stanford Brown iGEM team 2018 in my lab, and the students in 2 weeks built me a little stool and baked it, and it's not much to look at, but it gives you an idea that it really, it doesn't take much to build a structure. And as you can see, it's rated for an average size Stanford student. I got a little nervous, but I even stood on it to be filmed to prove to people it really is strong, and it was remarkable. They did this in 2 weeks. We've also looked a little bit at this co-culture, making the biocomposites with a photosynthetic organism, and here's some work that Chris did while my lab was shut down during COVID. The A there is just a little petri dish with cyanobacteria, filamentous cyanobacteria in it. And then B, what you've got is the dried cyanobacteria. I'm sorry, B is the hydrated, then inoculated with Aspergillus oryzae, which is a filamentous fungus. And C, you can see it overgrown with the fungus, and D, you can see the dried composites that we can use for materials testing. So again, it's taking advantage of this basic ecology that happens on planet Earth, taking it off planet. Here's a prototype that Chris was in the process of building in Cleveland when we were out there for a program review in September. And I just think it's remarkable, these things that he's building, and it just doesn't take that long. And so you can see these white patches of fungi on there. Obviously, there are Earth-based applications as well. That's not my day job, but they do like to know in this particular program, NASA Innovative Advanced Concepts, if there are applications for the Earth with the knowledge that many of the things they fund will never fly. And so we do have projects going with Azurmende, which is one of the top 50 restaurants in the world, and has gotten awards year after year for being the top sustainability restaurant. It's in the Basque region of Spain. No, I have not been there yet, but I'm planning to go in September. And so we've been working with them designing sustainability pods. So the idea is that whole structure would be made out of the fungal mycelia, the tables and the chairs and the whole deal, and see how much of this we could use and compost in the end. Both Chris and now there's a student at the Basque Culinary Institute working with me, have looked at making plates and chopsticks, forks, and all sorts of really cool things, finding things like used coffee grounds. Apparently those are very poor for sustainability rating, coffee grounds. I'm a tea drinker, so I don't really care as long as we don't get the coffee flavor into the plates, but they have done a little of that. And then Alessandro, the grad student at the Basque Culinary Institute, is working a little bit with advanced foods as well for Belle Cuisine. Other fungal-based foods are starting to be out in the market, even in the United States. So along with that, this material science, as I mentioned, organisms make materials, not just buying them, but they make, for example, chemistry, and I hardly need to say more there. And along with that, they also also make many of the pharmaceuticals that we use. And this is something that NASA had not really thought about a lot. We have an astronaut med list, and it's got 50 or 60 things you take with you, but the FDA doesn't tend to rate things for more than 2 to 3 years. They just don't bother. And if you're talking about a 2.5-year round trip mission with extra radiation, and there can be delays, and you have to load a lot of these things several months in advance, you could well need to know that you can get something for a much longer period of time. Now, what we have focused on in particular is a type of pharmaceutical. So the idea is that we want to do an on-demand system for just, making pharmaceuticals within 24 hours. And one of the big new classes of pharmaceuticals are peptide-based biologics. So these are proteins. Proteins are the easiest to make for a biologist, particularly for synthetic biologists, and they are the least stable. They tend to be only good for about 6 months, and these are often things that the pharmaceutical companies charge, literally an arm and a leg for. And so this is like a perfect conjunction of something that's easy for me to make and very unstable, but potentially very useful. And so we have had funding for a couple of years to work on a Master of Pharmacy. I have a proposal in to go into phase 2 on this. And it's a very simple idea where we have the Bacillus subtilis programmed to make whatever pharmaceutical, and you can dry, literally a gazillion Bacillus subtilis spores on a little piece of paper, so that's no problem, and then just take them with you dry. We've done that, LDEF has done that, and so on. And then when you want the drug, you add medium, and then you do a couple of purification steps, and you check for purity, and then you give it to the astronaut, and everyone's happy. I'm showing this in little tubes. We did a little prototype with the iGemmers and microfluidics that was the whole thing was about this big. The only problem is every time the students said, and then we use the pump too, and pumps are big. It's like, no, we can't use pumps. So I've pushed the new students into a syringe-based purification system. So again, this is just a schematic that went into our phase 2 proposal. But the idea is I want these down to a very tiny kit, which obviously could be useful for point of care on the servicing on the Earth, and we've talked a lot to DOD about being able to take things out into the field and so on. So again, imagine if you could just make a few doses on the spot, and everything's good. So that's really great. We also use light for material extraction, and we've had a big biomining project over the years. This is a mine not too far from Las Vegas, and these mining operations are big deals. And if you think about it, metals are heavy and bulky, and so if you don't have to carry a lot with you, so much the better. Here's some of the major elements that are used for space exploration, and here is some of the major elements that are used in life. But if you look at, there's quite a bit more overlap. So for example, there are major groups of organisms that use silicon, like the diatoms and the glass sponges, or aluminum is not very good, which is too bad because that's what my lords and masters at NASA really want me to do with aluminum. Chlorine, fluorine, many of these things, manganese, iron, at least some organisms do use. So the overlap is pretty good. And so if you think about it, the peptides and other molecules in your body that bind these metals do with incredible precision. So the hemoglobin in your body binds iron. You can't just say, well, maybe today I'll bind copper, or maybe today I'll bind fluorine. It doesn't work that way. You have to have incredibly low tolerance for error. Otherwise, we're not alive. And so if we can take advantage of the specificity of binding that the biological world has evolved, we can use that for biomining, for extracting metals, and certainly for separation. That's incredible. So we now have a database of natural and human-evolved peptides, short bits of proteins that will specifically bind, and we've actually gone through much of the periodic table. It's really quite amazing. So our proof of concept that NASA funded was to do an end-to-end recycling system. So here's an integrated circuit, and they do eventually go bad because of the radiation. So yeah, you could bring up lots of them, but if you're on a Mars trip, they may ultimately go bad. And so if you grind them up and then you make peptides that will bind the copper, and again, if you're wondering what sort of metals we use, I always tell people look at the back of the vitamin bottle because then you'll know the list of ones that humans need to have. You can grind them up, you can take these peptides that bind, for example, copper, which is what we did for proof of concept, and then we gave these peptides that had bound the copper to another colleague, Ram, who has a really cool plasma printer. And so the plasma printer basically vaporized the peptides with the copper, and he was able to print new interconnects, and so you have a closed circle. Now, that was great for this printing, but if you're really going to do something at scale for pulling out and separating metals, you need some kind of substrate that's better than just proteins. And so what we did ultimately was put together these two projects, the mycotecture and the biomining, and thought of using the fungal mycelia as the substrate itself. And so now we have a biological filter for metal extraction that itself is compostable and regeneratable and so on. And we have a paper out on that, and we have a patent that was filed 2.5 years ago that's working its way through the patent system. And so I'm really excited about that one. So let's get off material processing now, and I'll just whip you through a couple of other ideas. We send digital information. Think about it. Digital information is massless exploration. And that sounds crazy, but then you start to think through it. Now, I don't know how many biologists there are in the room, or anyone who's done any molecular biology, but great. So we got at least one. I'll keep 2. Excellent. 3. This is beginning to go like AA. You know, my name's Lynn, and I designed the sequence once. Anyway, you know that you sit there at a computer and you type out a sequence, or you cut and paste one, and you hit send, and your PI hits the credit card, and next thing you know, you've got a DNA sequence that's been synthesized. Now, think about that for a moment. You've taken digital information and turned it into biological information that then could be put in another organism or used directly in a cell-free system. And so you have broken that physical link. And if you can get someone across the hall to synthesize the DNA, why not bring a synthesizer with you to the moon or Mars? And then you send the sequences, which is literally massless exploration, and then you can just print them on site. And so, again, this sounds crazy, but it's not at all. It makes enormous sense. This is the first time in the history of life that we've been able to break that physical link from the origin of life to making something new. Now here's another big idea, nanotechnology. This young man, Simon Becchione, came to me when he was an undergraduate at Brown. He was the captain of our iGen team, and he said, I want to build a radio transmitter with synthetic biology. And I just looked at him and was like, I couldn't build a radio transmitter with a Radio Shack kit. By the way, for those of you who are curious, the SETI Institute, Search for Extraterrestrial Intelligence, defines intelligence as the ability to build a radio transmitter. And so when I'm with them, I always say, stupid life and proud of it, because I can't build a radio transmitter. Anyway, he wanted to do that, and obviously there was no way he was going to do that in 3 months, certainly not with me as an advisor. And so I talked him off the cliff, and he discovered in the literature that when there are mismatches in the DNA double helix from, instead of cytosine binding to guanine, if there's a cytosine-cytosine mismatch, you can actually get an interpolation of a silver atom. And once you know that, you suddenly realize you have the ability to use DNA to build a wire that's 1 atom in thickness. As it turns out, his more recent NMRs, he's now a postdoc. He's still working on this. We're still working together. He's doing fabulous stuff. He's now at NYU. It's actually 3 silver atoms per mismatch. But think about it. You now have this nanoscale precision of where you put each silver atom in this linear scale. He's now done DNA origami to do 3-dimensional work as well. But I mean, how cool is that? And by the way, if you have a thymine-thymine mismatch, you can bind mercury. And so we're also looking at other sorts of modified or novel bases to be able to see what we actually could interpolate into DNA. So this is the BioWires idea we've had. Now, why not use chemical energy to make electrical energy? And I remember remember the head of, it's now Armstrong, NASA Armstrong in Southern California asked me once, could you ever program an organism to make electricity? It's like, unless you were dead, you are doing that right now. No one in this room is eating to grow taller at this point. You are eating to metabolize, to fight entropy, and that includes creating electrical energy. That's your nervous system. An electric eel is just really good at storing that and releasing it all at once, but we all do that. So yes, and there are bacteria that do that as well. So that is certainly a potential. Now, why not push these environmental boundaries for life? And this gets back almost to my astrobiology roots, looking for the limits for life in the universe. If we could take an organism, for example, this Ocellatus that we want to make drugs, and lower the temperature, the optimal temperature for its activity, or for the fungi or whatever, it will take less power because we're closer to ambient temperature. Actually, the moon, it fluctuates both ways. And so the idea is to push the environmental boundaries for life with a project that the students started with years ago they call it the Hell Cell Project. We call it Synthetic Extremophiles and Polite Company, but I still love Hell Cell. And so we had 3 students for the summer who came to me and said, you know, we have these genes that will confer desiccation resistance, radiation resistance, the extremes of pH, heat, and cold. And why don't we take E. coli, Escherichia coli, this garden variety sort of wimpy bacterium that everyone uses as a sandbox because it's really easy to work with, and see if we can give it some superpowers. And by golly, in one summer they did all these, which I was shocked. This was 2012, and I mean, I was just amazed that 3 students were able to do this summer. And they all worked to some extent. The desiccation resistance one seemed to work the best. And there's the control, the blue bar, the E. coli undergoing desiccation resistance and showing very poor survival without with no additional genes, so empty plasmid. Tremulose, which is a sugar, should have conferred a lot of desiccation resistance, and you can see it did go up quite a bit. Glycine betaine is also commonly used by organisms for desiccation resistance. Manganese transporter we had not expected, but we were using it for other things, and mostly radiation protection, so they tried anyway, and remarkably it did well, so sometimes it's just worth trying what's around. So the answer is you can very quickly do this sort of thing. And so I've been showing that slide for now 10 years, and I finally have a postdoc who's picked this project back up. Not that we haven't had interest over the years, but we have funding proposed, and we have a postdoc working on it. And so he is doing the single gene approach, but what we also are doing is a different approach where we go out into nature, into extreme environments, and basically try to raid nature's hardware store without reading the labels, and then seeing what works, and then going back and reading it. I mean, it's almost like, you know, going to a hardware store and I need a battery and I don't know which one, so we'll just take them all and see which one works, and then we can find out how much that one cost and what the name was. It's sort of like that, and it works actually remarkably well. So we're doing a lot of that, and of course, laboratory evolution, because, I mean, in the end, Darwin always has the last say. We could actually start life de novo, and there are a lot of people working on that. There's a Build-A-Cell group. It's an NSF RCN that I'm part of the board for. We actually have an in-person meeting combined with a professional Build-A-Cell meeting in Minnesota in May, so people are interested. That should be on your radar screen. And Kate Adamala at the University of Minnesota, who's the PI on it, has been running a fabulous, for me Monday morning, for you it would be Monday noon, seminar series that virtually every week I am stunned at the people she finds. These, you know, everywhere from postdocs through faculty who are doing unbelievable work, who I've never heard of before, which is kind of stunning for me. But it really, I mean, the sort of work that's going on around the world is just amazing. And so, you know, there are people who are working on the membranes and the DNA and putting things together, including people in my group at Ames. Of course, we're more interested, the Ames funding is coming more for studying origin of life, how it actually happened, rather than building something new. But you know, both fields are are really important and can certainly play off each other. So I've given you a lot of wild and crazy ideas, and people say to me in NASA headquarters, yeah, these are wild, crazy ideas, but you know, we have to get people to the moon in 2 years, and we've got to get people to Mars in the next number of years. This is wild and crazy in the future. And so we realized it was time to test these ideas. And a few years ago, 2012, Senator Director Lynn came to me and said, I think the German Space Agency is going to give us the opportunity for secondary payload on their Europa mission. Do you have any ideas? This was Friday night. He said, we're in Germany. We're going to talk to them Monday morning. It's like, ouch, this is not the way flight experiments are supposed to work. And they said, oh, and by the way, we want something with synthetic biology. So I thought about our first iGEM project, which the students called Power Cell, and it was really envisioning a synthetic biology-enabled human colony. And so I said, well, we could do the Power Cell, and long and short is it ended up being selected as the secondary payload for this Eucropus mission, which was supposed to be Euglena crops in space. So the Germans were growing tomato plants with simulated urine and doing recycling. Sadly, they had communication problems, so they never got any data whatsoever from the mission, but I got plenty of data. So we're the secondary payload that in a way became the primary payload. And the Power Cell concept was exactly the one that I presented to you at the beginning of the lecture, if you remember back that far. The idea of having a photosynthetic organism as the interface between the raw materials on the planet and the production cells. And so we had, in the end, we didn't have enough time to really have enough spores of the algae. So what we did was we cheated a little bit. We used a lysate and freeze-dried that, so it would have been from the algae. And then activated the spores. And then we also tried some things like transformation and looked at promoter strength. This whole thing was done, I was given flight hardware that had flown before, which saved a lot of time and money. And basically, again, for those of you who worked in a biology lab, it was very similar to the form factor of a 96 well plate, except it was a 48 well plate. There were 4 rows of 12 wells. And the only things I was given, temperature control, we could have a little fluid flow, and there was basically a little tiny built-in spectrophotometer in every well. And so I had to design it around something that could do minimal fluid flow, and you could get the results visually. And so did it work? The answer is, oh, and I should have explained that the reason this was kind of interesting, this particular satellite, is it was to test the effects of gravity. So the idea is that there would be a certain period of time at the beginning where it wasn't spinning at all. So it would be mimicking microgravity like you would have on ISS. And then it would start to spin to mimic lunar gravity, which is a sixth of the Earth's gravity. And then 6 months later it would spin to a little faster, 1/3 G, to mimic the Martian gravity. So you could look at you could tease out the effect of gravity. I'm a bit less interested in gravity, more interested in radiation, but I always think to people, look, if there's an effect of gravity, we should know about it now. Maybe some things will work faster or better and low gravity, and maybe they won't, but let's find out now, and if they don't, fabulous, because then we can plow ahead in the lab without having to worry about that. But we really need to know. So this Power Cell thing worked fine, and there didn't actually seem to be a gravity effect. The transformation worked on the first run and not on the others, but we don't believe that was a gravity effect. It had to do with actually having sugar in there to make the cells competent when we get the fluid flow. And it turns out you cannot leave LB broth on the counter with sugar in it for 2 years, or it will start to turn brown and caramelizes. There's a Maillard reaction. Who knew? Okay, we now know. But anyway, we did get a transformation, and the promoter strength was sort of iffy because we expected a huge difference in the 2 different promoters, and we didn't see that difference. So I've given you a whole lot of sort of wild ideas about how life as a technology can enable human missions in the future, and again, I've been focusing on what we do. I work at NASA, National Aeronautics and Space Administration, and so we've also started to think a little bit about aeronautics, biofuels. The commercial industry is doing it on government, so we don't compete with the commercial industry, but you can immediately see that some of the materials that we're making could be useful for aircraft in the future. And of course, as I said, my background is the science, and a lot of what we're doing like looking at the limits for life, origin of life, and so on, you certainly could use a lot of this technology to answer some of these science questions. So I'd like to leave you with a few final thoughts, going back to the idea of green technology for a red planet. These are biological cycles on the Earth. Things like grass make the organic carbon, something like a cow might use it. We use products that gets recycled in the manure and the bacteria and so on, and everything's good to go. It's a really nice system. The tech path is not so green. We tend to take materials, we make something, we use it, we dispose of it, and we pollute. We're trying to make sure that that isn't always a one-way arrow with some of our technology. Once you're at ISS, the cost of going back and forth is incredibly high. And so some things we do actually make, use, and recycle. And the big one is water. The saying is, yesterday's coffee is tomorrow's coffee. I don't think I need to explain that to you. But that's not true, I've discovered, with things like laundry. We've had some proposals out now to develop really low-mass laundry systems. It's not true with a lot of the oxygen, with a lot of other things that we need. And you can resupply an ISS pretty easily, but it's going to be increasingly difficult as you go off planet. There's no biological cycle on the moon. We don't know for a fact that there isn't one on Mars today. Probably not, but we don't know that for a fact. And certainly we don't know in the past. So we not only should be careful, we have to. There are international treaties on this. We do have a lot of raw materials that would be potentially useful for in situ resource utilization. And maybe this will cut the costs then way down, because you won't need to go back and forth for everything. And so this is my vision, that we will take in situ resource utilization, we'll make things, we use them, and we'll recycle them off planet, cutting down these costs. Now, compared to people who are only focused on the Earth, maybe it sounds like I have a lot of constraints. And I do, but I don't have some of the constraints that you have. But I believe that these constraints will unleash creativity, and therefore force me to have, and my colleagues to have game-changing solutions that will be able to make huge, huge advances on Earth and beyond. And these game-changing advances will take us to the stars. So thank you very much. And obviously, there are a gazillion people who have been instrumental in this, and 10 years worth of IGEM teams. Again, thank you very much for your patience.
Thanks a lot for this.
You're quite welcome.
I know we are out of time, but if you have questions, the speaker will be here for 30 minutes if you don't have a run, but we can take one or two questions.
That was very interesting. I was wondering, like, when you are thinking about making the habitat, and this is probably because I don't know enough about Mars or the moon, like, don't you need to also make sure that it stays in place when you're growing it, or that there's always, it's not going to move from the location where you're just making it grow?
That's a really interesting question. You're worried about winds and so on.
Well, anything that may disrupt, right, the movement.
Right, so the atmospheric pressure is incredibly low in both places. So you do get some dust storms,
Right.
but it's not quite what you envision in the Martian. That being said, I'm sort of waffling because we have not actually included that, and you were the first person who's mentioned that. So I will go back and confirm that we will not have an issue. Otherwise, obviously, we will have to staple it down in some way.
Right. Yeah, I know about dust storms, but I don't know if that's like science
Yeah, I mean,
fiction or real.
Yeah, I mean, certainly there are dust storms, but it's not like in a dust storm here, because the atmosphere is so much thicker here.
Right.
Okay, so it should be okay, but thank you for bringing that up.
Is there any other question?
I saw you have Star Trek in the title. Replicator, is that one of your inspirations as well? I see a lot of that going on here. Little bits
Yes.
and pieces of being
Yes.
able to kind of simulate life, simulate production.
Yes, I mean, I would, yes, and that's sort of how I sold the bioprinting for a long time was, you know, could you imagine a replicator basically where you could print yourself a steak, and that's not, you know, a reality, not from my lab, from others. So yes, but the title came, although I understand legal is not totally happy with me using this title. That years ago, when we were trying to sell it, I was sent to talk to the NAC, the NASA Advisory Council, which at the time had Neil Armstrong on it, which is how I met him, and all sorts of fancy people. And the administrator can listen to them or not, but they are very, very high-powered people in the community. And I was asked to testify to the aeronautics subcommittee for some unknown reason. And I did, since this is going to be recording, there were 6 men in white shirts with pencil protectors. I mean, it was like my complete image of the aeronautics committee. And so I presented some of this. It was towards the beginning, so we really didn't have a lot of these cool projects going. And the chairman looked at me, and I thought he'd say, oh, that's, future, and he didn't. He said, well, so what? Wow me. And I thought, well, I did. So I took a deep breath and said, okay, we can do Star Trek. And that's where this whole sort of talk on here's a big idea, and here's a big idea, and here's a big idea came from. And over the last 10 years, I've sort of filled them in as I've started to work on all of them. I've sort of convinced myself. Yeah.
Hi, what does manufacturing look like? You mentioned it.
Good question. I think a lot of people are thinking sort of parallels the Earth with these bioreactors, where you make insulin and bioreactors, all these sorts of things. We have a few ideas on how they should be different off planet, but I think that's sort of the baseline. Now, something like these habitats don't need a bioreactor. So anyway, I think that tends to be the default, is thinking in terms of bioreactors. And now, obviously, you don't have to have the same scale up, at least not for a long period of time, and that's the problem, huge problem in the industries on the Earth, is this big scale up. But the level that we would need for the foreseeable future would be much lower. By the way, I've not only convinced NASA, I get calls from Space Force all the time, so I'm writing them. So I think that we've created a vision that's excited a lot of people.
I just with a quick show of hand, how many of you are from biomedical engineering? It will be the majority.
Oh, phew. Okay.
Mechanical engineering, we'll have few. Plant science, environmental science, I see 2 faculty.
Great, thank you for coming.
Yeah, so, yeah.
I would have talked more about algae if I'd known.
Okay, if, I know there's one more hand, and after that we'll wrap it up.
Yeah, I have a question. Have you guys thought about, because when you say, like, when you explain the habitats, it was going to be filled with the same oxygen that the plants were going to release. Have you guys thought about actually doing, because there's a point that actually it's going to be full, right? So you guys thought instead of like we bringing oxygen from here to like the moon or Mars, to just like storage the excess of oxygen, or just leave that so actually stores oxygen, so you guys don't have to take it from here to there?
Yes, so actually years and years and years ago, before this, I did a little project on that, because unlike what some of my colleagues think, the oxygen in photosynthesis comes from the water, right?
Yeah.
So plants split the water. And so that's a really good way to do that. And that's the idea, is that you would add a little water. Actually, fungi need very little water. So the idea is you have a little water would be enough to get the algae to then produce the oxygen and produce some more organic material, and then the fungi would eat that. Now, we've also, as a backup, are talking about having some nutrient-rich hydrogels on some of the drop stitching. So I didn't go into this, but if you inflate something, it ends up looking like a balloon, right?
Yeah.
But you don't want a habitat that's balloon, so the way you prevent it, you give it structure, is you have drop stitches from side to side, which is just a fancy way of saying pieces of, thread. And so you could then coat that with some nutrient solution and let the fungi eat that, and okay, it would be more up mass, but it would be trivial because they don't really take much. So we've sort of looked at both, but you really do need to regenerate the oxygen in any case, and I don't want to take it away from the humans. In fact, what I'd like to do for these habitats is have them there on a precursor mission. When you go on vacation, you don't want to arrive at a place and they say, oh, actually, we're going to build the hotel tonight or next week. You want the hotel already there, and I imagine the astronauts would too.
Thank you for all your questions. Thank you all for coming and for having the patience. specific.